Okay, hello everybody. So now we're going to start talking about cancer. And we're going to start by talking about specifically why medical researchers felt the need to develop targeted cancer therapies. Why wasn't the stuff we were using before good enough? So this is the third podcast in this sequence on modern cell biology and its relationship to targeted cancer therapy. There'll be one more video after this. But to start off this video, I wanted to talk about the original cancer treatments and why they ultimately have proved inadequate, why we wanted to really improve on them with these targeted therapies. So let's see, the original treatments, surgery to remove visible tumors, radiation to destroy visible tumors using high energy beams that can wreck the DNA of cells, hormone blockers, which stop the production of hormones for hormone-dependent cancers. There are a small number of cancers that are what are called sex hormone-dependent cancers. These are like breast cancer, prostate cancer, uterine cancer, ovarian cancer. These cancers are the reproductive organs. A lot of them tend to be hormone dependent, which means that they can't grow or reproduce without the presence of sex hormones. So that was something that was discovered actually quite a long time ago. And the old treatment for those kinds of cancers was castration. So removing the testicles in men or the ovaries in women, because that would stop the production of sex hormones and slow down the growth of those cancers. Nowadays, we don't actually have to really castrate people. We can chemically castrate them. That is, we can give people chemicals that stop their bodies from making sex hormones or stop the cancer cells from using the sex hormones. Those treatments actually tend to be pretty effective for the hormone-dependent cancers in that they can give essentially progression-free survival to people with those cancers of many, many years, typically anywhere from 2 to 5 years. And with relatively few side effects, since those hormone blockers don't have a lot of side effects. So that was the good news. The bad news is ultimately they become ineffective, and we'll talk about why. And then starting in the 1940s, and really discovered by accident, we found that chemicals that can stop cells from reproducing also can slow down the growth of cancer and sometimes even cure internal cancers. And that started the field of chemotherapy. Again, that discovery was kind of by accident. The original chemotherapy drug was nitrogen mustard, which was a biological poison that was used as a biowarfare agent back in World War I and then later on. And it was discovered kind of accidentally that this biowarfare agent actually was able to slow or reverse the growth of tumors in the body if people were exposed to enough of it, but not enough of it to kill them. And that led to the investigation into that weird phenomenon, led to the discovery that lots of different chemicals, which all had the ability to basically kill cells when they were trying to reproduce, were effective at treating various kinds of cancers. But why did we need to go there? Well, because quite frankly, surgery and radiation, which were the initial treatments for cancers, were inadequate and frequently did not lead to any kind of cure. So why was that? Well, I think most of you know that cancers, once they get going in the body, can spread through the body. That's what really makes them lethal, is they do what's called metastasis. which is, cancers that start in one part of the body, like say in the breast, can ultimately spread to other parts, to the lymph nodes, to the bones, to the brain. Some cancers spread to the lungs and to the liver. And, once cancers have started spreading around the body like that, you can't use surgery to effect a cure. And you might think, why not? I mean, if we just see where the tumors are, why don't we just remove all the tumors? Well, sometimes the tumors are growing. Once the cancer starts spreading, the tumors start growing in places that you really can't operate on safely. Like, a common area of metastasis is the brain. And, there are areas of the brain where you can have tumors growing which just aren't operable. If you tried to remove the tumor from that area of the brain, you'd remove a vital part of the brain and the person would die. Also, a lot of tumors spread to the lungs, and it's not really possible to remove part of a lung. So if you see tumors in the lungs, you kind of have to remove all of the lung in order to get rid of them. If you try just cutting out a tumor from part of the lung, you end up with a massive bleeding problem, in massive internal hemorrhage that can be fatal. Also, the fact that when cancer first starts to spread around, the cells that are spreading around are just like a few cells or small clumps of cells called microtumors, and those can't be seen on scans. So if you do a body scan on someone to look and see where their tumors are, you're going to miss some of the microtumors. They just won't be visible. There aren't enough cells there to really be visible on a scan. And so even if you removed everything that you could see, there would be a bunch of things you couldn't see, and you wouldn't know they were there until they grew into full-size tumors later on. And by then there would have been even more spread and more microtumors seated elsewhere. And you just can't keep removing pieces of the body. Eventually you run out of pieces. So surgery, once cancer begins to spread, surgery is not really good for effecting a cure. You can use it sometimes to make people more comfortable, but it's not going to affect a cure. And radiation is the same. It has the same drawback as surgery. You can only irradiate parts of the body where the tumors are visible, because radiation is very damaging to the normal cells of the body. So you can't irradiate the entire body. That would kill the person. Radiation is where you focus, concentrated beams of energy at cancer cells in order to destroy their DNA. But radiation also destroys the DNA of normal cells. And so, if you don't target it very precisely, you damage normal and healthy tissue. And once again, when cancer starts spreading, most of it is no longer visible. You've got microtumors and individual cells seated around the body, and you can't see those on scans, so you don't even know where to direct the radiation effectively. And so, if you just zap the tumors you can see, you're going to get other tumors growing very quickly that you can't see, and suddenly they pop up, and once again you've got the same issue that you have with surgery. You can't just keep zapping every part of the body. We talked about the hormone blockers. The only problem with hormone blockers is they only work on hormone-dependent cancers, which are just a few types of cancer, like cancer of the reproductive organs. And B, they lose effectiveness after a few years. And that's because of evolution. And we'll talk about that when we talk about the disadvantages of using drugs to treat cancer. Eventually, cancer cells, even the hormone-dependent ones, will evolve to the point where they are no longer hormone-dependent. And so those drugs will eventually stop working. But usually they give, for the sex organ cancers, they will give a few years at least of relatively symptom-free remission. And, that's a good thing. So they're still widely used for that. But because most cancers are not hormone dependent, at least not sex hormone dependent. And because radiation and surgery have limitations of, missing microtumors and individual cell spread, the standard surgery and radiation treatments are not usually curative. They're particularly not curative once the cancer has started to spread. If you catch a cancer very early, before it's really started to spread, you sometimes can cure it with surgery and radiation. But once it's really begun to spread and seeded microtumors around the body, then those will no longer affect a cure. And so because of all those limitations of the older treatments, we were looking for newer treatments and discovered accidentally in the 1940s, we found out that drugs that kill cells that are reproducing, in the act of reproducing, can often stop or slow cancer growth. And you can give those drugs into just the general circulation, and they can affect cancer cells and kill cancer cells anywhere in the body, because your blood circulates everywhere in the body. So even if you can't visualize all the microtumors and all the cells that have spread around, they can still be killed by standard chemotherapy drugs. So once cancer started to spread, chemotherapy was the only treatment that could effect a cure. And what chemotherapy is poisons, cell poisons that kill cells that are reproducing. Since cancer cells tend to reproduce rather quickly, they're a primary target for these chemotherapy drugs. So chemotherapy drugs can theoretically kill cancer cells wherever they are. And they preferentially often kill cancer cells because cancer cells are fast reproducing cells, and these are reproductive poisons. The only problem here is that, unfortunately, your cancer cells are not the only cells in your body that reproduce quickly. There are a lot of cells in your body that normally reproduce quickly. That's part of their normal activity, is they're just fast growing and fast reproducing cells, and those cells get massacred by standard chemotherapy drugs, which don't distinguish between cancer cells that reproduce fast and normal cells that are reproducing fast. So let's talk about the disadvantages of these chemotherapies, the drug-based therapies. That includes, by the way, the hormone blockers too, which are drugs. So because you have a lot of normal cells that reproduce fast, and because standard chemotherapy drugs don't distinguish between those cells and cancer cells, the side effects of normal chemotherapy treatment are pretty horrific. For example, the cells that normally reproduce very quickly in your body are your skin cells, the cells that line the inside of your digestive tract, so your mouth, your stomach, your large intestine and small intestine and rectum. All of those parts are lined with cells that are very fast reproducing cells. Your skin cells have fast reproducing cells. Your bone marrow and blood cells are fast reproducing cells. And so all of those types of cells get massacred by standard chemotherapy drugs, all of them. So, for example, standard chemotherapy drugs wipe out skin cells, and that can lead to you getting open wounds and sores that don't heal very well, and they become sites of infection. Also, you lose all types of blood cells, because those are fast reproducing cells. So you lose red blood cells, which are the cells that carry oxygen from your lungs to your tissues. And when your red cells drop, you become anemic, which means your tissues don't get enough oxygen to metabolize properly, to generate the energy that they need. Your cells generate energy by burning sugar and fat with oxygen, and if your cells don't get enough oxygen, they can't burn sugar and fat, which means they can't generate energy properly, and you experience that as feeling very, very tired and weak. and you have, significant organ dysfunction. Um, your white cells are massacred. They're also blood cells. Those are the cells of your immune system. So standard chemo wipes out your immune system, and that leaves you vulnerable to infections, since your immune system is the part of your body that protects you from infections. And like I said, the lining of your digestive tract, the entire internal lining of your digestive tract is made of fast reproducing cells. So that entire lining gets massacred by chemotherapy drugs. Your mouth, your stomach, your intestines. That leads to very severe gastrointestinal problems, vomiting, diarrhea, which can become continuous sores in your mouth as the tissue in your mouth starts coming apart. So it's all pretty terrible. When you're doing chemo, you feel weak, you feel tired. That's the anemia. You're vulnerable to infections and illness. Your skin develops cracks and sores, and your mouth develops sores. And then you get very sick to your stomach, you have trouble eating, you vomit a lot, and you have diarrhea a lot. All that contributes to dehydration and malnutrition, and so you just feel absolutely terrible. And this leads to a severe problem with cancer diagnoses. All cancers are easier to treat in their early stages than they are in their later stages, like before they've started spreading around the body versus after. But people tend to get their cancers diagnosed very late. And a lot of that is fear. People really fear a cancer diagnosis. Just the word cancer terrifies most people. But what I find interesting is I don't think that terror is due to the fact that cancer is a potentially fatal disease. There are other potentially fatal diseases that people don't seem to mind getting tested for, like cardiovascular disease. I think the real terror involved in cancer is not the disease itself, but the treatments. Pretty much everyone knows, because they've seen maybe family members go through it or friends go through it. I think almost everyone knows that cancer treatment makes you horribly sick. It's really awful. And I think people fear the treatment to some extent more than they do the disease. And so they resist going in and getting diagnosed with something that they fear might be cancer because they don't want to deal with the treatment for it. And that leads to late diagnosis, which leads to more mortality than there needs to be. And it's hard to blame people because the treatment is just god awful. If you can't just treat it with surgery and radiation, you're looking, or hormone blockers, you're looking at some miserable time. And there's other problems with chemotherapy too. So one thing that's interesting about chemotherapy and also radiation treatments is they're mutagenic. That means they cause mutations. Chemo drugs cause mutations, radiation causes mutations, and mutations are the cause of cancer. So when you treat one cancer with chemo drugs and radiation, those treatments can actually produce another cancer, a new cancer that may develop years later. And also the chemo drugs, because they're so toxic, chemotherapy can, in some cases, cause permanent organ damage. Like my best friend who had to go through chemo for his lymphoma, he had 9 months of chemotherapy. One of the drugs they used to treat his lymphoma was a chemotherapy drug which is effective but damages the heart. And so he was on that drug for 9 months, and it damaged his heart. And he didn't die of the cancer. He actually was cured of the cancer. But about 10 years later, he died of heart failure because of the damage that the drugs had done to his heart. So, in addition to making you feel sick while you're taking the treatment, chemo drugs can also permanently damage certain organs. And, so that can cause you problems long after the treatments are done. They can also lead to new cancers, which cause you problems long after the treatments are done. Chemo is also immunosuppressive. Again, we talked about how chemo kills white cells, which are fast reproducing cells, and those are the cells of your immune system. So chemo is immunosuppressive. When you're going through chemotherapy, your immune system does not work well. And that's a problem because, as we'll talk about when we talk about immunotherapies, your immune system is the primary system in your body that's designed to protect you against cancer. And even though it doesn't do a perfect job of that, even cancers that are fairly resistant to your immune system still does slow them down. So, when you go through chemo, the chemo itself slows down the cancer and may even stop it. But if it doesn't cure it, the immunosuppressive effect can lead to the cancers coming back and spreading even faster. But the real problem with chemo, all of those obviously are serious problems with chemo. One of the real problems, though, is, you may wonder why chemo ultimately fails to work. If it kills fast-growing cells, and cancer cells are fast-growing cells, why doesn't chemo cure you every time of your cancer? And there's a few reasons for that. But the main reason for that is evolution. So what chemo is cell poisons, and it poisons cells specifically when they're reproducing. Cells only spend part of their life cycle reproducing. But the cells of cancer, the cells that make up a tumor, are a diverse group of cells usually. They may all come from the same tissue, like breast cancer started as a mutated breast cancer cell or breast cell. But one of the oncogenes, one of the defects of cancer cells is they tend to be defective in their DNA repair systems. And that causes the cells to accumulate mutations very rapidly, which means that they can start changing rapidly. And so, if you have a tumor that's made up of, say, 1 million cells, even though all the cells ultimately came from the same mutated cell, the cells kept mutating, and because of their failing DNA repair systems. And, each time the cells reproduced, they mutated, but cells with different parent cells mutated in different ways. And like you have one cancer cell, it becomes 2 after it reproduces. But one of those may mutate in one way, and the other one may mutate in a different way. And so by the time you have 1 million cells, you have a very diverse group of cells with a lot of different mutations. And some of the mutations that they might pick up are mutations that make them resistant to chemotherapy drugs. And later on, we'll talk about different types of mutations that can make cells resistant to chemo. But often those mutations are in the cell reproduction pathway. So if you get more and more mutations in the cell reproduction pathway, a drug that targets cells that are reproducing may become less and less effective. That's why, by the way, when they give chemo for cancer, they don't usually just give you one drug. They give you several drugs. They might give you 3 or 4 different chemo drugs, which actually often target different parts of the reproductive pathway. And the reason for that is it's just too easy for cells to evolve resistance to one particular chemo drug. By the time you have a few 100,000 cells, it's almost guaranteed that any chemo drug you pick, you're going to have some cells that are just resistant to it because of the mutations they picked up. And so what they do to get around that problem is they use 3 or 4 drugs at the same time. So there's a chance when you use 3 or 4 different drugs that kill reproducing cells in different ways, there's a chance that none of them, particularly if you catch the cancer earlier, none of the cells have the right mutations to make them resistant to all of those drugs. So maybe they're resistant to 2 of them or to 3 of them, some of the cells, but none of the cells are resistant to all of them. And that means that there's a good likelihood that if you stay on that 4-drug regimen for 6 months or a year, you might be able to actually kill all the cancer cells and affect what they used to call a cure, and now they just call a permanent remission. They don't use the word cure anymore. So that's why, to get around this evolution problem by natural selection, whereby the poison selects for, it promotes the survival of cells that are resistant to the poison, which leads to large numbers of cells eventually having resistance to that poison. You can sometimes get around that problem with using a multiple drug regimen. But that doesn't always work. Sometimes, particularly if you catch a cancer in later stages and you have a lot more cancer cells in your body, which are a lot more mutated, there may be cells in there that are resistant to all of these chemo drugs. In which case, at first you'll see some kind of remission. The tumors may stop growing or may shrink for a little while, but then eventually the cells that are resistant to all of these drugs will start reproducing again, and tumors will regrow. And at that point, once the tumors regrow, they'll be full of cells that are resistant. So, doing more chemo with those same drugs won't help. It won't give you any additional survival. So what they may do at that point is switch to different drugs. But there aren't that many different classes of chemo drug, particularly the old style that just kill fast-growing cells. So, once you've gone through the top 4 different classes of them, you don't have that many left. So they can, try adding a few new types of drugs, and that may give you a little more, remission time. But again, eventually some of those cells will be resistant to those new drugs and tumors will come back. And at that point, they just don't have anything else to give you that your cancer cells won't be resistant to. Okay, so these are the disadvantages of the chemo drugs. This is why chemo fails, and to some extent why people are so resistant to getting diagnosed early with cancer, because they're so afraid of the treatments. And the treatments are pretty awful. When you're talking about classic chemotherapy, old style chemotherapy, the treatments are terrible. So overall, if we go back 50 years, the overall survival rate of cancer for people in this country was about 50%. That is, when I say survival rate, I'm talking about the 5-year survival rate, which means that, if you were diagnosed with cancer in the United States 50 years ago, there's statistically a 50-50 chance you would still be alive in 5 years. So half the people who were diagnosed wouldn't be and half would be. Some of that was due to the fact that the cancer was caught early and might be curable with surgery or radiation. And but a lot of it was just, a lot of the cures were due to, chemotherapy being effective for those cancers, even the classical chemotherapy. Now today, if you're diagnosed with cancer, your 5-year survival rate is over 60%. And a lot of that improvement is due to these discovery of new targeted therapies, which can either be used on their own or in addition to classical chemo drugs. And the point of developing the targeted therapies was just to have more ways of treating cancer, but also targeted therapy means giving somebody a treatment, a chemical, let's say, that will only kill the cancer cells and won't kill healthy cells. So won't cause the organ damage and the immunosuppression and the intestinal issues that classical chemo would cause. And so that would make the treatments less scary because they'd make you less sick. And also they might be, more effective. And at the very least, they would give us more, options of things to give somebody. So that was the idea behind looking for targeted therapies. But to find targeted therapies, we had to learn about oncogenes. Oncogenes are the genes that tend to be mutated in cancer cells. And we had to learn all about them. We had to learn about which genes they were, how the mutations changed their activity, which chemicals they interacted with in the cell, and how they interacted with them. And that was all a very heavy amount of basic biological research went into all of those discoveries. And it turns out that there's a lot of different oncogenes. For example, just in the cell reproduction pathways, which tend to be mutated in fact, they're mutated in pretty much all cancers. There isn't just like one gene involved in cell reproduction. There's dozens of genes involved in cell reproduction, and you can mutate any two of them pretty much and end up with a cell that could potentially become cancerous. But any of those 24 genes can be oncogenes. And so, some people have cancers where genes A and B are mutated, but some people have cancers where genes C and D are mutated. And in both cases, you end up with cells that reproduce too rapidly. But these people, they have different genes that are mutated. So even if their cancers are the same type, that is, they start in the same place, like maybe they're both breast cancers, but their breast cancers don't have the same oncogenes. They have different oncogenes, different reproductive genes that have been mutated. And so what we had to do was a lot of discovery work. We had to learn, which genes are the oncogenes, and then what do they do in the cell, and then which other chemicals do they interact with, which are involved in which pathway. That's a lot of work. We also wanted to find oncogenes that were common to several different types of cancer. So for example, a cell reproduction oncogene that was mutated in breast cancer, but also in prostate cancer, and also in skin cancer, and also in lung cancer. Could we find oncogenes that were more common in different types of cancers than others? So a lot of basic biology work there. All right, so let's talk about what targeted cancer therapy is. So targeted cancer therapy is usually chemically based treatments, chemical treatments that can destroy cancer cells without affecting normal cells. So again, that makes them different than classical chemo. Classical chemo kills cancer cells, but also kills a whole lot of normal cells too, because it's not specifically targeting. It targets any fast-growing cell, and there are lots of fast-growing cells in your body that normally are fast-growing cells. And so the question is, how can we target cancer cells specifically? How can we make a chemical that only affects cancer cells? It turns out there are 3 different ways of doing that. We can target the oncogenes. So, for example, if we have a tumor from a person, and we know which genes have been mutated in the tumor, and how they've been changed, and which chemicals those changed genes interact with to make them behave abnormally, we can then usually find, at least for some of those genes, we can find or develop chemicals that can shut the gene products off. Remember, genes code for proteins, and it's the proteins that are active in the cell. They're what causing, the bad proteins are what are causing the problem. So if you have bad proteins, for example, that are overactive, then if you just make chemicals that stick to those bad overactive proteins to make them stop working, then you can control the cancer that way. You're essentially shutting off that oncogene product. And remember from the previous video all the different ways that proteins are created and regulated, right? That we can control the creation and regulation. We've got epigenetic control of the creation of proteins, and then we've got post-translational control of the activity of proteins. So, for example, if there's a reproductive protein that's overactive in a cancer cell, if we know which protein it is and how it's been changed, we can probably develop some chemicals that can stick to that overactive protein and shut it down. And that would be a treatment that's specific for the oncogenic change here. So it's a treatment that sticks to the mutated form of the protein, but not to the normal form of the protein. So we try to design a chemical that'll stick to the mutated form of a protein, but not to the normal form. And then if we inject that chemical into the body and it can be absorbed by cells, it'll only affect the cancer cells because normal cells don't have the mutated form of the protein, but normal cells do. I'm sorry, but cancer cells do. So cancer cells and normal cells may absorb this chemical, but only the cancer cells will be affected by it. So that's a targeted therapy, but it involves our understanding of oncogenes inside the cell and our ability to develop chemicals that can stick to those oncogenes and change the way they work, and that are specific and that can be absorbed by cells. So that involves a lot of work. A lot of research goes into that. We're not talking about something that can be done the next day. If we haven't already discovered this, we're talking about years of discovery and research and then testing in order to come up with chemicals that can just affect the oncogene without affecting normal gene products, and that can be safely given to people without hurting them. Now, another way of targeting cancer cells is we can do what's called vector-targeted therapy. And what we do here is, this also involves, by the way, a discovery of some difference between cancer cells and normal cells. But here we're just looking at the surface of the cell, not the internal stuff. So it's easier, actually. We look at the surface. I think I mentioned that the surface of cancer cells is abnormal relative to normal cells. Some of the oncogene products in cancer cells are surface receptors, always. So cancer cell surfaces always have some differences from normal cell surfaces. In some cases, this difference is just too many of a certain type of receptor. In other cases, it's a mutated version of a receptor that doesn't have the same normal shape as the normal version. So if we can identify some, an abnormal protein on the surface of cancer cells, we can design chemicals or cells or, essentially polymers, which are groups of chemicals that form into shapes, to bond specifically to those abnormal proteins on the surface of the cancer cells. So, and then we can attach like oncogenic therapies, or we can attach just regular cell poisons like regular chemo to these vectors. Vectors are the modified cells or chemicals that stick specifically to abnormal chemicals on the cell surface. And if we attach then poisons to the vectors, cell poisons, then we can use the vector to do our targeting for us. We can use the vector to bring the poisons directly to the cancer cells. And if the vectors are designed correctly, they'll stick to these abnormal surface chemicals on the cancer cells, but they won't stick to normal chemicals on the surface of normal cells. So they will bring whatever poison we choose to use, whether it's a targeted poison or an untargeted poison, just a general poison that would kill any cell, we can bring it directly to the cancer cell using this vector. So we attach the poison to the vector, and then the vector will attach to some abnormal chemical on the surface of the cancer cell and bring the poison directly into the cancer cell. Another way to do targeted therapy is we've learned fairly recently that the internal environment and the external environment of cancer cells is different than most normal cells. For example, the inside of a cancer cell, the environment inside the cell, tends to be very acidic compared to normal cells. And the outside of the cancer cell, the area around the cancer cell, tends to be also acidic compared to normal cells, but also it tends to have much lower oxygen levels than the environment around normal cells. And there's other differences too. So it's possible to design, for example, a cell poison that is only active in an acidic environment, but it's inactive in a normal environment, normal pH environment, like a neutral environment. So if you have a poison like that, where the poison's active in an acidic environment but not active in a neutral or basic environment, if you expose your body to that poison, presumably that poison will only be activated when it enters the cancer cells, which are very acidic inside, and it will not be active when it enters normal cells. So it's targeted in the sense of, we're not trying to make it so it only bonds to cancer cells. It can bond to any cell and get into any cell, but it's only active inside cancer cells. Similar in that way to oncogenic targeted therapy, which is only active inside cancer cells because it's bonding to the abnormal forms of proteins in the cell. Here, we're just using the general environment. So we don't even need to know, to do micro-targeted therapy, environmentally targeted therapy, we don't even need to know oncogenes. We don't need to identify them. We just need to identify how the environment inside and outside cancer cells is different than normal. Recently, they've developed experimentally a drug that's activated when oxygen levels are low. And so this drug tends to become active when it gets to a tumor, because the oxygen levels inside the tumor tend to be very low compared to the rest of the body. And so the drug only gets activated when it enters the tumor, and then it becomes active, and then it can actually kill cells. So that limits it to killing cells in the tumor, which is the environment where it's active. Now, in reality, we've never developed a perfect microenvironment targeted therapy. There's always some activation in normal tissue. So there's always some side effects of these, but getting better at it. Okay, so I wanted to talk a little more about the vector targeted therapies, and I'll show you a picture here. Let me shrink this a little bit. Okay, So these are vector-targeted treatments, and here the red dots represent poisons, which can be attached or enclosed in various kinds of vectors. And there's all different kinds of vectors we've got now. For example, one of the more common vectors is what's called a monoclonal antibody, which is an antibody is an immune system chemical that's designed to stick to the surface of abnormal cells. We can design antibodies to stick to the surface of cancer cells, taking advantage of these abnormal receptors on the surface of cancer cells. We can design antibodies that will only stick to those abnormal receptors and won't stick to the normal versions. And then we can attach some kind of cell poison, which can be targeted or untargeted, to these monoclonal antibodies. And if we've done this right, the monoclonal antibody will only stick to the cancer cells, so we'll only deliver these poisons into the cancer cells. They won't be delivered to normal cells because these antibodies won't stick to the normal cells. We've also developed a number of what are called nanopolymers. And nanopolymers are basically combinations of chemicals that form like cages or bubbles. And inside these cages or bubbles that are made of many different molecules stuck together, we can insert a whole bunch of different poisons because there's more space. The problem with these antibodies is there's only room to attach like one poison to an antibody. But these nanoparticles are actually much bigger than the antibodies. And they include things like liposomes, which are little fat bubbles, or DNA cages, which are literally, cages made of short segments of DNA and RNA. But there's enough space inside these things where you can put in multiple poisons. So the advantage is you can use multiple poisons to poison cells instead of just one. And if you've done this correctly, the DNA cages and liposomes can be made with chemicals on their surfaces that only bond to these abnormal receptors on the tumors. And so they'll attach themselves to the tumor cells, and then they'll get absorbed by the tumor cells. And once they're absorbed, they'll get broken down inside the cell, and they'll release their poisons to kill the cells. We can also use engineered cells as nanoparticles. Like we can use bacteria that have been killed or viruses that have been killed. The thing about bacteria and viruses is they already have chemicals on their surface that let them bond to specific types of cells. And we can fill them up, even though they're dead, we can fill them up with poisons. And they'll stick to the cells they normally stick to, and then when the cells absorb them, again, they'll release the poisons and kill the cells. So these are various kinds of targeted treatments, which are vector targeted. So the poison can be any kind of cell poison. It can be a targeted one or an untargeted one. Sometimes the poisons, for example, are actually radioactive atoms, which release high-intensity radiation that doesn't travel very far. So when a cell absorbs these vectors with these radioactive particles, the particles will release radiation that only kills that cell. But it's a very non-specific radiation. It would kill any cell that it was absorbed by. But if it's attached properly to a vector, it'll only kill the cells that the vector bonds to. So if we've designed these vectors correctly, we can attach them to any kind of poison, to a specific poison or a non-specific poison, and we can kill just the cancer cells if we've done this all right. So these things have become, fairly widely used now, these vector-targeted poisons. These other things, the oncogenic targets and the microenvironment targets, here we don't need to use vectors because theoretically only cancer cells will be affected by oncogenic targeted therapies and only cancer cells will be affected by microenvironment targeted therapies. So vectors are unnecessary for these. Vectors are necessary if we're going to use just a general poison. If we're going to try to kill the cancer with a general cheap cell poison, we would need a vector targeted therapy so we didn't kill healthy cells. Okay, but all of these are forms of targeted therapy. Okay, I'm going to focus on some oncogenic targeted therapies because those are kind of the most interesting of the newer therapies. So oncogenic targeted therapies rely on our understanding of oncogenes, which genes are mutated in a cancer cell. So which genes can we mutate to give ourselves a cancer cell? And it's never one gene, right? There is no single gene you can mutate in any way that would give you cancer. In order to turn a normal cell into a cancerous cell, you have to mutate at least 6 or 7 or 8 different genes. Or maybe only 4 or 5 different genes, but then you'd need to mutate a couple of different genes involved in some of those different pathways. Now, if you remember before about how we regulate, gene products, we can regulate gene expression, we can regulate protein activity. There are some oncogenes that are what are called anti-cancer genes. So cells all have, because cancer has been a problem as long as there have been, vertebrates, basically, which is a long, long time. Your genome, your DNA, contains some genes that are anti-cancer genes. They're specifically designed to prevent a cell from becoming cancerous. Okay, so some of the anti-cancer genes are like DNA repair genes. Genes that normally create proteins that fix damaged DNA. Because remember, cancer is a mutational disease. It's a disease caused by damaged DNA. So DNA repair genes are naturally anti-cancer genes. They repair DNA damage. So they repair some of that DNA damage that they're repairing is oncogenic damage. So they're repairing oncogenes and turning them back into normal genes. So that's an anti-cancer gene, is a DNA repair gene. You also have cell reproductive genes, which are normally designed to stop cell reproduction. Okay, those are anti-cancer. Not the genes that are designed to encourage cell reproduction. Those are different type. And then there are genes involved in cell suicide. It turns out, a lot of different cell suicide genes. And these are genes that are normally activated by abnormalities in the cell, which could be cancerous. For example, when there's significant DNA damage, or when cells are starting to move around and locate themselves in the wrong part of the body. Those kinds of conditions are designed to activate suicide genes in the cell, which kill the cell so that it can't go on and become a cancer cell. So these are anti-cancer genes. And then you have pro-cancer genes, which are genes that, if their activity were increased, they would make cancer more likely. So for example, genes that encourage cell reproduction, genes that stimulate cell reproduction, are pro-cancer genes, because if you make them even more active than normal, if you make them super active, they cause cells to reproduce super fast, which is one of the hallmarks of cancer. So another type of pro-cancer gene is, for example, a gene that makes cells that increases cells' ability to move around. Because cancer cells, one of the ways they spread through the body is they regain their ability to move around. most of your cells, once you reach childhood, they lose their ability to move around because they're now located in the organ where they're supposed to be, and you don't want them to leave that organ. The liver cells are all in the liver, and the skin cells are all in the skin, and you don't want them to go other places. So, as you age, your genes that are involved in cell motility, the ability of cells to change their location, those tend to get shut off. But those are pro-cancer genes, because if you turn them on, if you activate them, then they let cells move around, and that encourages cancer. So bottom line is you've got a number of pro-cancer oncogenes, which are become real oncogenes when they're mutated to make them more active. And then you've got anti-cancer oncogenes, which can contribute to cancer if they're mutated to make them less active. So the anti-cancer oncogenes, when they're mutated to make them less active, they stimulate cancer development. The pro-cancer oncogenes, if they're mutated to make them more active, that stimulates cancer development. But again, you need to have a lot of these genes in different pathways that contribute to different processes in the cell, mutated to get yourself an actual cancer cell. So let's talk about the different types of oncogenes. Oncogenes are genes that, if they're mutated in particular ways, they contribute to the growth and development of cancer. I'll put myself here. So here are the pro-cancer types of oncogenes. So genes involved in cell growth and reproduction, specifically genes that stimulate cell growth and reproduction. If you make those more active, then cells can reproduce abnormally fast, and that creates tumors. So those are pro-cancer genes, oncogenes. Two of the ones that are very commonly mutated in cancers are what's called the epidermal growth hormone receptor gene, which bond to a hormone called epidermal growth hormone, which stimulates the growth and reproduction of a lot of different cell types. And then the RAS gene, which also stimulates the growth and reproduction of a lot of different cell types. So if you make too much of these, or if you make them so that they can't be inactivated, then they'll contribute to cells reproducing too fast. Again, genes involved in cell motility, like there's a MET gene, which is important for cell motility, and the TORC genes, which are important for cell motility. Remember, for each different process here, there isn't just one gene. There's a number of genes involved in cell reproduction, a number of different genes involved in cell motility. And so you can theoretically mutate any of them. And if you mutate them in the right way or the wrong way, then they become an oncogene. So a mutation in like the MET gene or the TORC genes that make them more active is also oncogenic, because now the cell has an easier time moving around and spreading. There are genes involved in angiogenesis. Angiogenesis is the formation of new blood vessels. When tumors grow, you need to create new blood vessels to feed the tumor cells, otherwise they all die. And so cells in the tumors activate they usually have activated genes like the VEGF gene, which is the vascular epidermal growth factor gene. And that gets turned on in these cells, which allow them to develop new blood vessels. It allows some of the cells in the tumor to actually turn into blood vessel cells. And that brings in new blood to the tumor to feed the tumor cells. So if you reactivate or stimulate these genes, then That makes cancer more likely because it allows tumors to grow without dying from lack of food. Now, in fact, pretty much all cancers that become really dangerous, life-threatening cancers, have the ability to evade the immune system. We'll talk more about that in the final video when we talk about immunotherapy. But there's a number of different genes that, if they're overactivated or reactivated, they can protect the cell from attack by the immune system. And those are pro-cancer genes, because if the immune system recognizes and destroys a cancer cell, the cancer can't grow. So cancer cells that can protect themselves by reactivating genes that protect them from the white cells, that's pro-cancer. And then genes that can create channels that eliminate chemotherapy drugs. For example, there are certain energy-using channels that cells can make that can push some chemotherapy drugs out of the cell. So if you give somebody chemo with these drugs and they have these channels, then as fast as the chemo drugs get into the cell, they'll be pushed out of the cell again. And then that'll allow the cell to survive the chemo. So those are pro-cancer genes, because they allow the cell to survive treatments for the cancer. And then there's the anti-cancer genes. We've talked about these, the genes involved in DNA repair, like the BRCA genes. People with BRCA1 or BRCA2 mutations, they have mutations in these genes which normally produce proteins that repair damaged DNA. If you have mutation in these genes, you can't repair your damaged DNA. And so the cells accumulate mutations very rapidly. And once you accumulate enough mutations in the right genes or the wrong genes, then you've got cancer. So these genes involved in DNA repair, if you inactivate them, that makes cancer more likely and makes it more quickly. It also makes the cancers evolve more rapidly so that they become resistant to like chemo drugs. And then the genes involved in cell death, in cell suicide. If you mutate those genes, then cancer cells will not kill themselves when they normally would. That is, when they sense that they're in the wrong location in the body, or that they're reproducing too fast, or that they're otherwise, or that their DNA is too messed up. Normally, all that would trigger cells to activate their suicide genes and kill themselves. And that would stop the growth of the cancer, the development of the cancer. But if those genes are mutated so that they no longer work, then cells can't kill themselves when they should, and that allows them to develop into cancer cells. Pretty much all the cancers we found where we've identified the oncogenes, there are some cell death genes that are mutated in pretty much 100% of cancers. But there are different cells, and there are different genes involved in different types of suicide, like the p53 gene, the BCL2 gene, even the RAS gene is involved in some types of suicide. So like 50% of all cancers we've identified have a mutation in the p53 gene, which inactivates it. So if you inactivate these, because these are anti-cancer genes, if you inactivate these, that makes cancer more likely. If you hyperactivate these, make them more active, that makes cancer more likely. So for these, you would need to deactivate them. For these, you would need to hyperactivate them. Now, you may wonder, how do you hyperactivate a gene? Well, in most cases, it's one of two things. You either create the protein which has now lost its inhibitory regulatory site. Remember last video we talked about how a lot of proteins have inhibitor sites, where if another chemical bonds to that site, it stops the protein from doing its thing, from being active, or it reduces its activity temporarily. So there are some of these pro-cancer oncogenes, which if they're mutated in the regulatory part of the protein that bonds to an inhibitor, so that they can no longer bond to the inhibitor, then essentially the protein is now always active. Before, the cell was able to regulate it. It was able to make it inactive for part of the time and active for part of the time. But if you destroy its inhibitory site, now the protein is always active and can't be deactivated. So one very common way that these genes are made more active is they have mutations in their inhibitory sites so that they no longer can bond to their inhibitor. But there are other ways to make genes more active. For example, at the epigenetic level, if you, for example, mutate the cell so that the repressor for their gene is inhibited, and you can no longer make the repressor, or it's mutated so that it no longer works, then you can no longer reduce the transcription of that gene. Remember, repressors actually reduce the transcription of a gene, the turning of the gene into messenger RNA and into protein. So that can be controlled at the genetic level by making repressors that prevent, like RNA polymerase, from opening up that gene. But if you mutate the repressor so that it doesn't work, then now the gene is always active and you're always making that protein. You can even sometimes mutate the epigenetic alteration of histone proteins that lock up areas of DNA. And if you mutate the histone protein in a certain way, you might not be able to lock up certain portions of DNA, which means that the genes there are always being made, even when they shouldn't be anymore. So you can make genes more active at the epigenetic level by allowing the cell to transcribe them when the cell normally wouldn't transcribe them. Or you can make them more active by a mutation in the gene that destroys their inhibitory regulatory site, the regulatory site on the protein that bonds to an inhibitor. So in that case, the gene is still working, but now you can't make it stop working. So there are different ways of actually getting a mutation that increases the activity of a protein. And of course, there are a lot of ways of getting a mutation that decreases the activity of protein. The most basic way is just a mutation that destroys the active site of the protein so that it can't bond to the chemical it normally bonds to. But in order to develop an oncogenic treatment, a targeted oncogenic treatment, we have to know which of these proteins is mutated and in what way. For example, if we have too much of, say, a gene product that's involved in cell reproduction, how is it mutated? Is it mutated to destroy its inhibitory site? Or is the repressor for it mutated? Or is the histone mutated so that that gene is available for transcription where normally it wouldn't be? We need to know that, because knowing that tells us which treatment we need to design to be effective in reversing that change. We're trying to reverse that change. But to reverse it, we need to know what the change actually is, which thing was changed to make the cell behave this way. It's actually harder to deal with the anti-cancer genes, because anti-cancer genes, you've inactivated the protein. You've probably got a mutation that's destroyed the active site of the protein. Well, the only way to fix that directly would be to repair the DNA itself so that you could now make proteins with their active site restored. There's usually no way to add an outside chemical to a cell that can restore an active site that's been destroyed. So dealing with these is actually much harder than dealing with these. Because here we can usually add a chemical that can reverse the oncogenic effect. Here, it's much tougher to reverse the oncogenic effect. Although we have found some ways. Some of them are very clever. Like I'll tell you about one very clever way to reverse the effect of defective DNA repair protein. Okay, so I'm going to talk now, to finish off the video, I'm going to talk about some of the treatments that we've developed that are targeted treatments, oncogenically targeted treatments. So, for example, remember I said that one of the ways that cells become cancerous is when they activate genes that can protect them from immune system attack. Well, one of the major genes that you can activate to protect yourself from immune system attack is called the PDL1 gene. So the PDL1 gene puts a chemical on the surface of cells called PDL1, which bonds to a chemical on the surface of immune cells and deactivates the immune cell. So when an immune cell touches the cell with PDL1, it gets deactivated. So the easiest way this is relatively straightforward to inhibit this effect. What we do is we create chemicals that stick to PDL1 and prevent it from bonding to the receptor on the white cells. So let's see. I may not have a pic. I don't have a picture of that here. But this is pretty straightforward. We've got what are called checkpoint inhibitor chemicals, which stick to PDL1 and prevent the PDL1 from bonding to the receptor on the white cell that deactivates the white cell. And so that keeps the white cells working and allows them to recognize and attack the cancer cells, even the ones that have made this white cell deactivating chemical. Okay, another chemical that's commonly made on cancer cells is a mutated version of epidermal growth hormone receptor, which is a receptor that bonds to a growth hormone which stimulates the reproduction of the cell. So a lot of cancer cells, they make too much of this receptor, and they make a version that's harder to turn off, so that it tends to be always on, and there's too much of it, and so it causes the cells to reproduce too quickly because it's an overactive growth hormone receptor. But we've developed a chemical, this was actually the first targeted chemical, called Herceptin. And whoops, that's the wrong one. Where's my Herceptin? Here it is. Okay, so we developed this chemical called Herceptin. And what Herceptin does is it bonds to this abnormal growth hormone receptor, and it prevents it from combining with another growth hormone receptor. So for these growth hormone receptors to work, to signal the cell to reproduce, you actually have to get two of them bonding together. It's when two of the growth hormone receptors bond together that signals the cell to reproduce. Normally, that only happens when they're bound to the growth hormone. That's when they tend to stick together. And when two of them stick together, that signals the cell to reproduce. But what Herceptin does is it gets between them. So when these receptors are bound to growth hormone, epidermal growth hormone, they try to get together, but the Herceptin keeps them apart. And so they can't stick together, and so they can't activate cell reproduction. Okay, so this chemical that gets between these growth hormone receptors and keeps them apart, that was the first targeted chemotherapy called Herceptin, and it's still being used today. It works for a number of different cancers because there's a number of different types of cancer that are stimulated by having too much of this growth hormone receptor, epidermal growth hormone receptor. Number of cancers are stimulated to reproduce by having too much of that receptor. But it can only stimulate them if two of those receptors can bond to each other, and Herceptin prevents that. So that's a targeted therapy because it's really most effective on cells that overproduce growth hormone receptor and produce abnormal versions that get together more easily with other receptors. And Herceptin is designed specifically to stick to those. So it's a targeted therapy, it's fairly effective, and it's used even today as not usually by itself, but in combination with other therapies. And it does it causes longer remissions. So when you give it in combination with other therapies, people tend to get longer remissions than they would just using the older therapies by themselves. Another targeted therapy that was developed very recently is a RAS therapy. Now, RAS is a protein that stimulates cell reproduction. And the version that's mutated in most cancer cells is called KRAS. That's the oncogenic or mutated version of RAS. And the thing about KRAS is what makes KRAS bad is KRAS proteins, they stick together, they can't be shut off. And they stick together very easily. This is another one of those cases. It turns out there's a lot of these in your body. Proteins that are only active when they combine with other proteins. Right? So when they're by themselves, they're not active, but when they dimerize, when they combine with one other protein that's similar to them, then they become active. And RAS is like that. So here's the RAS protein. And a normal RAS protein bonds to a chemical called GTP, which it then breaks down to a chemical called GDP. And the GDP form doesn't stimulate, it doesn't dimerize, and it doesn't stimulate reproduction, but the GTP form does stimulate reproduction. And the mutated version of RAS, it bonds to GTP and never lets it go or converts it to GDP. And it bonds to GTP very, very tightly. So essentially, it's always active and always on. But it needs to combine with another RAS protein in order to become active. Now, you would think that it would be pretty easy to stop this chemical. All we'd have to do is design a chemical that could bond to the active site where the RAS essentially bonds to another RAS to become an active complex. But the problem is the RAS protein is very smooth like a Ken doll. And where it combines with another RAS protein is a very smooth area. It's not a typical active site which has nooks and crannies and some specific shape. It doesn't have any specific shape. It's a very smooth area that just attaches itself to another smooth area of another RAS protein. And so it wasn't possible to make an inhibitor. We couldn't make an inhibitor where it bonds to GTP because it bonds so tightly to GTP, it essentially never lets it go, and there was no way to get an inhibitor into that site. And where it bonds to another RAS protein is so smooth that it wasn't possible to design a specific inhibitor for it. because any inhibitor that would stick to it would stick to a whole bunch of other things too. Basically, any smooth part of any chemical. So just recently, we finally developed an inhibitor for the RAS protein. Again, this protein stimulates the reproduction of cells. It's necessary for the reproduction of lots of different cells. And what's interesting about this inhibitor is it doesn't directly bond to RAS. It actually bonds to a different chemical called CYPA, which is also a very smooth chemical with just a little notch in the middle. And this inhibitor, called daraxonrasib, it bonds to this smooth part in the middle and makes it not smooth, makes it bumpy. And that allows this smooth CYPA chemical to attach itself to the smooth part of the RAS chemical, which just has this little notch in it. So this chemical is what's called a linker. It links the smooth CYPA to the smooth part of the RAS. And once those things are linked together, then this area is no longer the right shape to allow RAS to bond to another RAS protein. So you can't get the dimerization. You can't get the two RASs bonding together and becoming an active complex. And if you can't get two RASs together, you can't get the stimulation of the cell reproduction from RAS. So this was a very clever workaround to what had been a really difficult inhibitor problem. RAS was considered uninhibitable because of how tightly it bound to the stimulator chemical GTP and how smooth the dimerization area was. It was considered an undruggable chemical, but through the cleverness of human ingenuity and a little computer-aided design, we were able to develop this chemical that allowed us to link the smooth part of RAS to the smooth part of another chemical that's naturally in the cell. And so to create a smooth area that was no longer the right shape to allow RAS to dimerize and become active. And this chemical, which was just approved last year, is great at treating pancreatic cancer, which some of you may know is one of the hardest cancers to treat. It's very unresponsive to most chemotherapy drugs. And most people who are diagnosed with pancreatic cancer, have a life expectancy that's measured in just a few months. A lot of people diagnosed with pancreatic cancer only live a few weeks after diagnosis, one of the most rapidly fatal cancers. And there have not been any real effective treatments for it up until now. It turns out that daraxonrasib increases dramatically the survival of times of people with pancreatic cancer. And I'm talking about dramatically. The average survival time goes from like 4 to 6 months, which is the average, to 12 to 24 months. Using a targeted therapy that doesn't cause many side effects. There aren't many side effects of this chemical because, again, it only, it has some side effects because it can affect the active version of RAS in normal cells and slow down the reproduction of normal cells that need to reproduce. But it is a reversible effect, and it's got a much weaker effect in normal cells than it does in cancer cells. So although there are side effects to this treatment, they're usually tolerable, not as bad as conventional chemotherapy side effects in most cases. And the survival increases are very dramatic for people, especially with pancreatic cancer. By the way, 80% of pancreatic cancers have this RAS mutation. So it works with most people who have pancreatic cancer, which is fantastic. Okay, another really clever targeted treatment. Remember I said that targeting inhibitory genes that normally prevent cancer. Targeting them is more difficult because those genes have been shut off by the oncogenic mutation. And if the gene's been shut off, it's pretty hard to reactivate it. If its active site has been destroyed through the mutation, it's impossible to reactivate it. So it's very hard normally to develop a treatment that targets tumor suppressor genes. But we have developed one recently, and that's called a PARP inhibitor. So one of the tumor suppressor genes you have is genes that repair damaged DNA, right? Because those prevent the development of cancer. And even once a cancer has developed, those genes can slow down the mutations in those cancers so that the cancer develops more slowly and becomes resistant to chemo more slowly than a normal cancer cell would. So a lot of people, for example, with cancer have BRCA1 and BRCA2 mutations in their cancer cells. And these are genes that normally create proteins that repair damaged DNA. And they repair severely damaged DNA, what are called double-stranded breaks in DNA, which is a severe form of DNA damage. So if you have mutations in those genes, your cells become unable to repair this bad form of DNA damage, and they accumulate mutations very, very quickly that can cause dramatic changes in the cell. What PARP inhibitors do, it's very clever because you can't, once these genes have been damaged, you can't restore them. BRCA1 and BRCA2 mutations are not reversible in any way. You can't bring back these dead proteins, essentially. So we go the other way here. This treatment, which is a targeted treatment, what it does, instead of trying to bring back the DNA repair system, which it can't do, what you do is you make it even worse. So you give these cells a chemical that makes their DNA repair systems even worse than they already are. In fact, you make them so bad that now the cell can't repair anything, and it dies pretty quickly from DNA damage. So this chemical is taking advantage of these mutations by actually making them worse than they already are. So you take cells that are unable to repair these bad mutations, but are still able to repair minor DNA mutations. So PARP mutations are minor single-stranded DNA breaks. They're minor mutations which are normally fairly easy for cells to repair. What PARP inhibitors do is they convert minor DNA damage into major damage. So where cells have single-stranded breaks and they send in PARP proteins to repair the single-stranded breaks, the PARP inhibitors then cause those proteins to fail so that they turn the single-stranded breaks into double-stranded breaks. And since single-stranded breaks are much more common than double-stranded breaks, you start getting catastrophic DNA damage all over the genome. And that causes the cell to die from DNA damage pretty fast. So this is a very clever way of dealing with this problem where, when you inhibit anti-cancer genes, it's very hard to reverse the inhibition. Here, we're not trying to reverse the inhibition. We're actually trying to make it worse. We're trying to make it so bad that the cell just dies. And that's what PARP inhibitors do. So PARP inhibitors are now being used to treat people who have like BRCA1 and BRCA2 mutations, which are mutations in the genes that allow cells to repair unusual but very serious DNA damage. Okay, we also, as I mentioned earlier, we have like sex hormone blockers, which are forms of chemical castration, where instead of actually castrating someone with a sex hormone dependent cancer, we chemically castrate them. We give them chemicals that prevent them from creating sex hormones or using sex hormones in order to stop the growth of sex hormone dependent cancers. So again, all of these are targeted therapies. They only work in cells that already have the oncogenic mutations. For example, the PARP inhibitors, if a normal cell absorbs a PARP inhibitor, it's true that it actually turns its single-stranded breaks into double-stranded breaks, but it also has active double-stranded repair systems so that it can repair those double-stranded breaks so that they don't kill the cell. So PARP inhibitors, although they do have some side effects, they only will actually kill a cell with a BRCA1 or BRCA2 mutation. They won't kill a normal cell, although they will increase the DNA damage of that cell, but then the cell will be able to repair itself. Okay, so studying oncogenes, learning about different kinds of oncogenes and how they affect cells and ways to reverse their effects, or in this case, to enhance the effect to lethality. We've been able to develop some much more targeted treatments. And all of these treatments have one thing in common, and that's that they have fewer side effects than conventional chemotherapy drugs because they're more targeted. They tend to only affect cancer cells, or they affect cancer cells much more severely than they affect normal cells. And so they have at least that advantage. And the other advantage is we can combine these with each other or with conventional chemotherapy drugs to create, even longer remissions. It gives us more treatments that we can use to create longer remissions in cancer. And that's why not only have the overall survival rates for cancer gone up more recently because of the targeted therapies, but what's called the progression-free survival time, which is essentially the amount of remission time you get from a treatment, that's gone up very dramatically for a lot of different kinds of cancer recently. In a lot of cases from months to years, where you may not have cured the cancer, but you can keep somebody feeling pretty good because their tumors aren't growing or spreading during that period of time. And that's also due to these new targeted therapies. So the bottom line is they're helping. They haven't given us the end-all cure-all for cancer. And we'll talk more about why that is next video. But they've definitely helped. Helped increase the overall survival rate modestly, and helped increase the progression-free survival rates fairly dramatically compared to the conventional chemotherapies. Okay, so that's it for video 3, which focused on targeted therapy, particularly oncogenically targeted therapies. The last video, video 4, will focus on immunotherapies, therapies that involve getting your immune system to attack your cancer. All right, so I'll see you in video 4.